Now, good day and you're welcome back. This is the second part of our series where we are discussing viruses. And like I asked before, what are viruses? What makes them special? Why do we study them? We've already earlier established that the study of viruses is called virology. And to continue our discourse on viruses, we say that within a viral particle, you cannot have both DNA and RNA. You're going to have just one, either a DNA molecule or an RNA molecule. And this is important to note. Now, it's also important to note that viruses exist in two major states or phases. Two major states or phases are where viruses exist. And viruses exist in an extracellular phase or an intracellular phase. Extracellular or intracellular. Now, remember that viruses are problematic because of the difficulty of being able to classify them into a group and the fact that they are not really living things, but they are acellular entities. Now, when we say extracellular phase or intracellular phase, what do we mean? We realize that when the virus is existing outside of a living cell, it is within its extracellular phase at that time. And when it's within its extracellular phase at that time, the virus possesses very few enzymes and it cannot replicate. It cannot reproduce in the extracellular phase. Now, the second phase that viruses exist in is the intracellular phase. And when you have viruses existing in the intracellular phase, they are different. And in the intracellular phase, the viruses are able to replicate. Now, the intracellular phase means that the viruses are now inhabiting a living organism. They are staying within a living cell. And when you have the viruses within a living cell, they are able to replicate. And how do they replicate? They replicate using the machinery of the host. That is all the structures, the sugars, the carbohydrates, the proteins, the RNA and DNA that are present within the host. The viruses use up all these materials and use them to make many copies of themselves. So this is both the extracellular and the intracellular phase of the viruses. Now, it's important to note that in order to replicate or reproduce viruses, or let's use the word cultivate viruses in the laboratory, those viruses need to be injected. They need to be inoculated into living cells. There are many ways through which viruses can be replicated and can be cultivated within the laboratory. We sometimes use the chick embryo to cultivate viruses, and you inject such viruses into embryo of chicks or chickens, and then they replicate within that embryo. This is important to note and important to see. And like I said before, the viruses can be enveloped or they may be without envelope. Now, when these viruses are being cultivated, there is also methods, assays through which the viruses can be purified after they have been cultivated within these living organisms. They are purified using various different types of enzymatic assays, and they are purified according to their weight. How heavy the viral particle is determines the method of purification of viruses. We're still going to do videos where we're going to discuss about the purification of viruses and the various ways in which this is achieved. Now, the life cycle of viruses. We'll end this class discussing the life cycle of viruses. There are 5 major steps in the life cycle of viruses. The first step is the attachment to the host cell. Before a virus is able to, go through its whole life cycle, one, it has to attach itself to the host cell. That is the living cell to which it wants to gain entrance and replicate. It will attach itself into that living cell, one. Secondly, it will gain entry into the living cell. Many a times, the virus is injected into the living cell and it leaves its capsid or its coat behind, and just the DNA or the RNA molecule is injected into the living cell. Well, now, when this virus gets into the living cell, the third part, the first part is the entry, is the attachment to living cell, entry into living cell. Then the third step in the life cycle of viruses is the synthesis of viral nucleic matter. Viral nucleic acids are synthesized within the living host. Like we said earlier, viruses usually use the machinery of their living host to make many copies of themselves. So when the virus is injected into this living host, it uses up the machinery of the living host and uses it to make many copies of itself. This is important. Now, the fourth part or the fourth step is the self-assembly of viruses within the living host. Now, why the initial virus is within the living host? It will make many copies of itself, and then it would, assemble protein coats around all these new viral particles that have been produced within the living cell that is a host for the virus. It will assemble new capsids, new protein coats around that living host. And then the fifth and final part of the life cycle is that there's the release of the viruses from the living host, the release of the viruses from the living host. This is very important to note. 5 steps in the life cycle of viruses. That's the attachment to the host cell, entry into the host cell, synthesis of viral nucleic acid and proteins, then assembly of viruses within the host cell, then the release of the viruses within the host cell. Now we'll end this class by discussing just 3 ways in which viruses differ from other non-living, other living organisms. Remember, the viruses are neither here nor there. We call them acellular entities. So one is that they are simple acellular entities. One, now they have either the DNA or the RNA. Meanwhile, most living cells have both DNA and RNA, but for the viruses, it is either going to have a DNA or it will have an RNA. And then, lastly, viruses are unable to replicate outside a living host. They only replicate when they find themselves within another living host. Thank you for listening to this lecture. Please remember to like our content, share our content, and we'll all have a wonderful day. Thank you.